So we are going to talk about today deviations from Mendel's law of independent assortment. And that actually led to discovery of genetic recombination. So our learning outcome today is basically the discovery of genetic recombination and what these deviations really meant. Whether these deviations simply disapproved what Mendel discovered, or they discovered that Mendel was absolutely right, but there are still some more secrets to be discovered when we talk about genes residing on the chromosomes. So if you remember, we talked about Mendelian yellow and wrinkled when we crossed with green and round. All the F1 was yellow round. When we self-cross this, what we discover is this is what the gametes are for this parent. And the probability of these gametes in case of law of independent assortment was equal. It was 25% each, or you can also say 1/4. Okay, 1 in 4 will be this one, 1 in 4 will be this one, 1 in 4 this one. However, if you carefully look, what is very clear that these two, this one and this one, so we write them parental gametes. We write them parental because they are the gametes which were present in the original parents, not the F1. Remember, the original parents from which we drove the F1. So original parents had capital Y small r, small y capital R, and then we had non-parental which were not part of the original parents. So the ratio between parental and non-parental gametes in here was 50% each, or so this together 50 and the non-parentals were also 50. So basically they were equal. Now, when T.H. Morgan, the man who introduced fruit flies to study genetics in the labs, they set up crosses, and they were, and the Mendel was already rediscovered in 1904, 1905. So what Morgan's lab was doing, Morgan's lab was, having fly mutants. So those of you who don't know, the organism is fruit fly. And yeah, Qasim?
सर जो आपने टेबल बनाया था क्लास में जिसमें 16 कॉम्बिनेशन है पुनर्स्केयर में तो वो सर इससे बने थे या वो जो
या
प्रोडक्ट्स के
16 कॉम्बिनेशन 16 कॉम्बिनेशंस इसी से बनेंगे ना। जब आप इनको 4 गैमीट्स इधर 4 इधर होंगे, सेल्फ करोगे। मैं सिर्फ एक टाइम पे गैमीट्स की रेशियोज आपको बता रहा हूं, ठीक है। अगर आप y कैपिटल R, y R, y कैपिटल स्मॉल r एंड स्मॉल y, इसके 4 ही गैमीट्स आप इस तरफ बनाओगे तो 16 कॉम्बिनेशंस वही 9331 रेशियो आ जाएगी। क्लियर?
जीनोटाइपिक, यस सर।
So why he opted to bring in fruit fly was because as compared to pea plants, fruit fly life cycle was much shorter. So from egg laying back to an adult, it takes total 10 days. We start from egg, time point 0, and we go, the whole life cycle is complete in 10 days. So generation time is much smaller. Okay. And Morgan lab, they discovered mutants. The normal body color of fly was brown, but the mutant they discovered, a homozygous recessive mutant, was black. And similarly, Morgan's lab had a wing phenotype, which was, so the normal wing is very well expanded, a structure like this with veins and then some cross veins. But and the normal wing phenotype was dominant over a homozygous recessive trait. The mutant phenotype they named was vestigial. Vestigial was just a rudimentary wing like this, a very clear, but this was also homozygous recessive. So normal wing was dominant. So we will write something like this is the normal wing and the small letters for vestigial phenotype. Brown, capital B, and black was this. So when they set up homozygous, when they set up monohybrid cross, they followed perfectly the law of segregation, in which they had these black flies crossed with normal flies. Normal means brown. They found all normal flies which were brown, wild type phenotype. And when they set up a cross of F1 male with F1 female, they found 3:1 ratio of black versus brown. Same was the case for vestigial. So they had vestigial fly crossed with normal wings. Everything was normal here in F1, but when they set up cross between males and females, F1 males and females, they found 3:1 vestigial versus normal wings. Ji Amna?
Sir, here, like, how in F1 you did the cross between the heterozygous and the dominant and the homozygous recessive, right?
No. So
Isn't
let's say
the
we have black,
mm-hmm.
and so
Oh, it's only
F1
one
will be
trait. Sorry, it's only one
this.
trait. I was mixing the two traits together.
Yeah. And when you have males with this phenotype, you will get 3:1. 2 browns will be this one and 1 black. And same is the case for vestigial. Now, what they did next was they said, okay, let's take the black vestigial and cross them with normal flies, normal wings, and normal body color. What will be the phenotypes? Sorry, genotypes.
Small b.
Small b, VB.
VJ.
Very good. And
Black, sorry, capital B.
Capital VG, capital VG.
Capital VG.
we can also write them like plus over plus. This represents homolog chromosomes which carry normal alleles of B, and then vestigial plus over plus for capital VG, the two homolog chromosomes. This one is plus, plus means wild type, normal. And here you can simply write minus over minus, and then this minus minus represents homozygous recessive on two homologs, and then minus minus for vestigial homozygous recessive. But let's go. What will be F1?
Capital B, small b, capital VG, small v.
This will be F1.
Yes.
Clear?
Yes, sir.
Yes, sir.
Okay. Now they set up a test cross here. And test cross, what will be the test cross?
Small b, small b.
Small b, small b, small
small
bg,
b.
small bg.
Small v,
Very
small
good.
v.
Now this is very important point that in fruit flies, we always use, whenever we set up a test cross and we have to see the organization of these genes on a chromosome, I mean, this is a fact we know in 21st century. I fail to understand how Morgan Lab may have learned all this early on. There is always, we use tester male. So this will be homozygous recessive tester male. And on this side, they use female. And soon we are going to see why we saw, why we are using females. Now, according to what we saw here, when you set up a test cross or whatever in F1, let's say self-crossing, or if we say we set up a test cross here, not selfing, let's forget it. We set up test cross. The gametic combination will be 25%, which means 50% parental, 50% non-parental. So 25% each from F1 individual. Is it true? Now, if we set up a test cross, what we expect is
25%, 25%.
we expect 25% each. And what will be parental? Who will help me to write down the parental gametes?
Is it capital B, capital VG?
Capital B, capital VG, and
Small b, small v.
where what will be non-parental?
Capital B, small
small
b.
vg.
Small b, capital VG.
Small b, capital VG.
And how much should be the expectations from, if we say gametes, how much percentage we should get?
25% each.
So these two, this and this one, we said these are non-parental. and these two are
Parental.
parentals. And we said each class should be 25% each. Isn't this according to Mendel?
Yes, sir.
When they counted, they found that the capital B VG or small b VG, which were parentals, they both were like 965 and 944, and these were parentals. And the non-parentals, they discovered they were sorry, this should be small b capital VG and capital B small. This should be small, okay? Small VG. They found that these numbers were like 206 and 185. This was a puzzle. How can this happen that these are way too high in number and these are too small numbers? They are neither, I mean, this is a big puzzle, isn't it?
Yes.
They said, let's
Ji,
do
sir.
another experiment. Here we used black vestigial as one parent and normal wing and body color on the other. Let's do an experiment in which we use black body color, normal wings, and brown, which is normal body color, vestigial. Went to F1, everything was normal. Set up a test cross, which was indeed this one, and again counted the F2, the gametic combinations. So who will help me write the parental gametes and non-parental gametes now?
Small b,
Hmm?
small b.
Sir, capital B, small vg, small vg.
Parentals.
Capital B, small v. Yes.
Capital
Capital
B,
B,
small
capital
b.
VG.
Let him speak, who was earlier. Please
Sir,
go on.
math, sorry.
Ji, aap please, Ahmed.
Sir, black is, I'm sorry. Sir, black is small b and normal wings are capital V.
Very
Capital
good.
VG.
What will be
So,
other
sir,
parentals?
small b and capital V.
So this is
And
one
sir, capital
parental.
B and
What will
small
be the
VG.
second parental?
Sir, capital B and small VG.
Very good. So these are basically the parentals we are talking about. Okay, not the F1. And what will be non-parental?
Capital B, capital VG, and small b and small VG.
What they discovered, I mean, they thought maybe we should get again 25% because we are now bringing mutant alleles from different parents. But what they discovered, the numbers were again more or less, the parentals were 960, 985, 206, and something like 190 or 180, whatever. Which means the results they are getting, they are not artifact. They are absolutely right. They are both more or less the same numbers. Now this was a puzzle, and what they learned that, in Mendel's case, where we had I write Mendel on this side and Morgan on that side. So if I had capital Y, small y, R, and set up a test cross, actually these genes are residing on independent chromosomes. Okay. And then the second chromosome maybe is, which carries round or wrinkled allele, is something like this. So when meiosis takes place, the homologs are going to this chromosome and this, they get duplicated. Okay. Due to DNA replication, this homolog gets also duplicated. This one as well. Its sister chromatids will look like this. Its sister chromatid will look like this. Now, during meiosis, when this goes to opposite poles in meiosis 1, we learned that independent assortment means that two different factors, which today we know as genes, two different factors which affect or impact two independent traits, they are going to segregate independently. They are going to assort independently. So which means this capital Y, when the gametes are formed, I'm not going to repeat meiosis again. We have done this multiple times in previous 3 lectures. So this capital Y may end up with capital R, or capital Y after reductional division and complete meiosis may end up with this one, small y with this one, and small y with. So there's equal chance, and this is because they are on independent chromosomes. This is what Morgan's lab described, that this is the only explanation. That's how we can get 25% each. And they said, what if capital Y and R and capital Y, small y and R, they small r, if they reside on the same chromosome, so after DNA replication, what will happen? We will get, the sister chromatids will look like this. The other homolog will look like this. In the gametes, we should get this one can go with either this one or with this one with this one. So essentially what we will get is capital Y. Sorry, I was wrong. So when they will go to opposite poles, so the gametes will be either capital Y, because these sister chromatids will be separated in the after reductional divisions. So after reductional division, these two homologs, so this will go to one pole, this will go to other pole, and after completion of meiosis, we will get gametes, this one or this one. So which means if they reside on the same chromosome here, if they reside on the same chromosome, we should get 50% each parental. We should not get any non-parental because they are on the same chromosome. Yeah, there was a question.
Yes, sir. I had a question that previously when you used the test cross, you used the homozygous recessive, and you used that tester male, and you said there was a specific reason for using like a male one. Why was that?
Yeah, I come to that point. I'll come to that point. Okay. So now what we have learned, if yellow round are residing on independent chromosomes, then this is what we should get, and Mendel was absolutely right. But if they reside on the same chromosome, we should get, you know, only 50% each parentals if they are residing on same chromosome. But Morgan's lab discovered that, look, we are not getting 50% each. Our genes are definitely not lying on independent chromosomes because they had the tricks in genetics. They could follow their mutants, and they knew that their genes are residing on the same chromosome. But they postulated that if they are residing on the same chromosome, black and vestigial, according to Mendelian law of independent assortment, we should get 50% each, only parentals and no non-parental. But what we are getting is non-parental, whether we bring in mutations from two independent parents or from the same parent. Many people working in Morgan's lab tried to tackle this problem. And if you have watched the lecture carefully, you might have seen I mentioned a sophomore student, Stratevant. Morgan's lab was, I would say, a high energy lab. Everybody there had this in those times, 1920 to 1940. They discovered so much, which we know. So our understanding of genes, chromosomes, chromosome inheritance, all these things. So they not only proved Mendel again and again, but they found deviations from Mendelian law, and they added more to Mendel's discoveries, which hold true even today. So nobody wanted to tackle this problem, what is happening. Sophomore student came in, and Stratevant was given, this problem. After setting up all these crosses, getting these numbers, what Stratevant did, he said, okay, let's take all the non-parentals over total progeny and calculate the percent new combinations. Isn't it new combination? This is not present in parents, either of the two parents. So these non-parentals, these ones, they are not present in either of the two parents. So how are we getting them? So what it did, he said, okay, let's add up these numbers. 206, 185, 206 plus 185, then 944 plus 985 plus 206 plus 185 into 100, and the number he got was close to 17%. In both the cases, whether it was this cross or this cross. So these new combinations were 17%, instead of, if they reside on the same chromosome, we should have received only parental 50-50 and 0 here. These 17% led Stratevant to discover something what we know today as genetic map. Organization of genes on linear chromosomes. What Stratevant said, he said, black and vestigial and the wild type alleles. He said the distance, the physical distance between two independent genes, one is black, the other is vestigial. He said this distance is resulting in recombination. They are physically exchanging, the chromosomes are physically exchanging fragments. And he explained this, that he said, okay, if we are, imagine we are doing this cross, homozygous recessive black and normal wings. This is your F1, okay? At DNA replication, so sorry, the two sister chromatids will look like this. So what Stratevant suggested, that during meiosis, recombination is taking place, and this recombination is determining how much percentage of non-parental we will get. And this percentage is directly proportional to the physical distance between the two loci, two specific genes. Greater the distance between black and vestigial, higher the percentage of recombination or consequent non-parentals. Shorter the distance between black and vestigial, less recombination, consequently we'll have less number of non-parentals. So he explained it like this. He said, okay, we will get, biggest majority we will get parentals, which is this one, the 4 gametes I'm going to make. And then sister chromatids, they exchange reciprocally, a crisscross structure is formed, and as a result, due to this physical exchange, we get now, so this small b recombines with homolog chromosome and we get capital VG. And then in another, as a result of this recombination, the other gamete will be capital B and small vg, capital B, small vg. These two are non-parentals. This one is parental and this one is parental. So Stratevant suggested that during gamete formation, during meiosis, what happens when chromosomes, homolog chromosomes get fully duplicated, the sister chromatids are formed. Let's say we have black, 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 and vestigial, vestigial and normal for vestigial. He said before the reductional division, before the homologs go to opposite poles, what happens, they come closer to each other and they physically exchange the fragments. Let's say you had black here and capital VG here, black and capital VG, and here you have small b VG and its sister chromatid small b VG. So as a result of this recombination, please mute your mic. So who is this?
Really?
Who was this? Would you mind sharing your name? Who was this? In Zoom, I can track everything. So I like honesty. Nothing will happen, don't worry. So you are not going to share your name? Hello. The one who said sorry, please share your name. I want to talk to you directly.
Sir, I think he left the meeting.
Pardon?
सर जो आई थिंक था ना वो जो भी था उन्होंने मीटिंग छोड़ दी है तो
So this recombination, this discovery led to discovery of genetic recombination. Abdullah, are you there?
Yes, sir, I am.
I want name of the student.
Okay.
So what we learned today, we learned that Mendel was not at all wrong. Mendel's laws, they hold true even today. But what Morgan's lab added to Mendelian discovery was that the factors which Mendel discovered, they indeed, and by that time we call these factors genes. So genes, they reside on chromosomes. And if genes reside on same chromosome, based on the distance between the two genes, they will undergo genetic recombination in meiosis.
Sir, I have a question.
G, please go ahead.
So, sir, basically, does that mean कि जितना distance होगा, does it mean कि like बार-बार cross करवाने से वो distance increase करता जाएगा?
नहीं, जीन अल्लाह ने सेट कर दिए हैं, जहां पे हैं वो वहीं पे रहेंगे.
तो सर like जो 17% हमारा आया था, उसमें से the student proposed that the difference between the gene small b and small vg, उनका distance 70.17 है, right?
Yeah, so I explain you what that 17% is, that distance. So this 17% means percent recombination. It means if there are 100 meiotic events, if 100 independent meiosis took place, there will be 17 gametes which will be non-parentals, or what we call today recombinants.
So, sir, that is not referring to the distance between the genes, right?
That is referring to the distance between the genes. And they name this physical distance, they call this centimorgan. Centimorgan, to honor their mentor, T.H. Morgan. 17 centimorgans means black and vestigial. The distance, physical distance is 17. So 17 centimorgan or percent recombination, or there's another thing, recombination frequency, and frequency can be maximum 1, so it will be 0.17. So this recombination during meiosis is telling you the distance, the physical distance. Let's say the physical distance between black and vestigial is 20 centimorgans. So you will get 20% non-parentals. Did I answer your question?
Sir, I'm just like confused about this one fact that why do we like genes तो कहीं पे भी हो सकते हैं ना on that length of a chromosome. So
No.
why the genes correspond?
Genes are present, black gene or whatever the normal body color is present on a specific, very specific position on chromosome in flies, which is So position of a gene on a chromosome is called locus, and plural is loci. So position of genes is fixed on chromosomes. Let's say vestigial is here. So in every Drosophila melanogaster, it will be here, except mutant. Clear?
Yes, sir. And sir, one more question.
Yeah.
Mendel performed multiple experiments with the pea plants, right?
Yeah.
So why never he found that 17% wala?
Because I think I explained it in my lecture. Because Mendel was extremely lucky. So he studied 7 traits. P has 7 chromosomes, and what we learned after Mendel's rediscovery and characterization of P chromosomes, that each one of that trait Mendel studied, it was on an independent chromosome. Okay, let's go and take quiz.
Sir?
Next lecture, we are going to prove, because this is what we learned today through genetic recombination. In next lecture, we are going to prove, and not we, Morgan's lab proved that genes indeed reside on the chromosome. Because next lecture is very interesting if you go and watch. That's about sex chromosomes and the sex-linked inheritance, which basically proved that genes indeed reside on the chromosome. They brought the proof. Yeah, there was a question.
Sir, I just had a small question regarding the chromosomes.
Yeah.
Like, this confuses me that, we have like 23 chromosomes, right? And like 46 when 23 homologs. So like, are all the chromosomes the same? Or like, say the first chromosome and the second chromosome both have R's, but like for the first one R actually shows and the second one it doesn't show. Are they the same or not?
Beta, this is chromosome 1, chromosome 2. I'm just trying. You go on like drawing 46 chromosomes.
Okay.
Genes on the chromosome 1, and this is homolog, identical pair of them, and this is homolog of this. Okay?
Okay.
Let's say there's a gene X here, gene X will be here too. And this gene X, if it is a single copy gene, it will not be present on any of the other 22 chromosomes pairs. It will only be here.
Okay.
Let's say there's a gene B here, on homolog, exactly same locus will be B. If it is also single copy gene, it will not at all be present on chromosome 1 or 3 or 4 or 5. And then along the length of the chromosome, because this is a hundreds of kilobases of it, here will be a different gene, here will be a different gene, here will be a different gene. So on each chromosome, there will be different sets of independent genes, which may not be present on other chromosomes if they are single copy genes. Clear?
Sir, what do you mean by single copy genes?
So there are genes in our cells which exist as single copy, which means one copy here and the one here, just once on the homologs. And if I say X is not a single copy gene, let's say X has 2 copies, it means X may have, and again, we will know the position through it may have a same X on chromosome 3, or on the same chromosome here, or maybe on this arm. So those genes are called multi-copy genes, more than one. But in Mendelian genetics and in Morgan genetics, at 101 level, we just talk about single copy genes. Did I answer your question?
Yes, sir. Thank you.
Sir, can I ask a question?
Okay.
Sir, so basically towards the end of your recorded lecture, you talked about those two different like traits like B and VG that are like 50 cm apart. So you said in that case, we will be confused at whether they are on independent chromosomes or not. Could you please like explain it a little further? Because I didn't really get it.
Very good question. She's talking about, in the lecture I said, if we know that black and vestigial are on the same chromosome, but the non-parental versus parental, and we again get 25% each. How is it possible? We know they are on the same chromosome. Such thing can happen if black is on one side of the chromosome arm or of the centromere and the other is here. The distance between black and vestigial is so great that it spans over chromosome, over centromere. So in such a case, we may get confused because, you know, this arm will always show a recombination like this. Not always, but, you know, the distance is so big that we get a picture like independent assortment, like genes are on independent chromosomes. They are not on independent chromosomes. They are just on the same chromosome, but distance is too huge, spanning over centromere. Clear?
So sir, if someone wants to find out that whether like he wants to confirm whether these B and VG are on another chromosome or what, so how can he distinguish between that?
Very good question. So you will simply find another mutation on, let's say, you know, this is chromosome 2. If you know this is chromosome 2 of fruit fly, you will find another mutation, let's say Y, cross it with vestigial, Y, cross it with black. If you find recombination between Y and vestigial and Y and black, you will say they are on the same chromosome, because genetic recombination can only take place when you are on the same chromosome. Yeah, there was a question about tester male, and I said, I promise I'll get back to it. When we are looking for these recombination percentage, where was that? I said tester male. I wrote it somewhere.
It is
Here
in
it is written,
this.
tester male. When we want to study recombination, the reason we use males as homozygous recessive parent and heterozygous mother. So this sign is mother, female in biology, and this sign is male. What we know, and Morgan's lab knew this, that females, they show zero recombination. Males in fruit fly, they show zero recombination, no recombination at all. And it's only females which show recombination. So when you are going to map a gene, when you are going to, let's say, map gene A, M, N, P along chromosome, same chromosome, you will use females and tester. So male will be tester always.
And how did they figure it out that only like mother gametes like
when you set up crosses, let's say you took male here, you just took reciprocal cross, you took
Okay,
black.
yeah, I get it.
You got it? So let's go. Yeah.
Are we going to have a quiz right now?
Yes, I announced like 10 minutes ago, but questions
Oh.
are coming, so I don't hesitate taking questions. Please go on.
जी सर, इसके बाद मेरी वो class है, so that's why I wanted to
So
just
let's
have
go
the
to quiz. Let's log into.
I've just published a quiz. You can log into LMS. It will be available. And सिर्फ एक सवाल है, 2 मिनट है आपके पास.
It's just a short, tiny question.
Sir, if I have another very good question, can the quiz be postponed?
Pardon?
No, because I have published the quiz right now.
If I have another very good question, can the quiz be postponed?
No, you can write me an email. I will answer you there.
Oh.
Don't waste time. Okay. Hurry up. Allah Hafiz. Good luck with your quiz.